The overall goal of this experiment is to screen reactions using a stop-flow microtubing reactor platform that combines elements from both continuous flow and batch reactors.
We invented this technology to overcome the problem that the continuous microtubing flow system is not efficient for small-scale reaction as well as reaction screening.
The difference between our stop-flow microtubing reactor system and continuous microtubing reactor system is that there are two valves before and after each microtubing in our system.
The main advantages of this technique are that it is green, safe, easy to use for reaction screening, and more efficient than conventional batch reactors for gas-involved and light-promoted transformation.
To prepare a stop-flow microtubing reactor, wrap 300 centimeters of high-purity PFA tubing into a narrow ring. Secure the tubing with zip ties and attach a shutoff valve to each end.
It's easy to construct an SFMT reactor, therefore we just prepare an SFMT reaction for each condition to be screened.
Connect the reactor to a 20 psi back pressure regulator and a low-pressure T-fitting connected to an acetylene gas tank via a needle valve. Submerge the end of the back pressure regulator outfit in acetone. Next, place 58.5 milligrams of 4-iodoanisole in a 10 milliliter round-bottom flask with a stir bar. Add to the flask 8.5 milligrams of bis-triphenylphosphine palladium 2-chloride, 1 milligram of copper 1 iodide,